Well, hello everyone. So we'll start by introducing ourselves since you're going to hear from several of us. So my name is Mariela. I'm a grad student and I study biochemistry here at Cornell. And now Ariane.
Hi, my name is Ariana. I study genetics at Cornell and I study breast cancer. And Nada Viviana.
Hi everyone, my name's Viviana. I'm a combined veterinary PhD student at Cornell and I study immunology. And next will be Dariana.
Hi everyone, I'm an undergraduate student at Cornell and I study biology and society. And now Morgan.
Hi everyone, I'm Morgan. I am a PhD student here at Cornell studying soil biogeochemistry.
Perfect. So now that we have introduced ourselves, so welcome to Snip Genetics. And today we will guide you through the wondrous world of gene editing.
So as you all know, every living organism has DNA. Since it's the blueprint of life, DNA is the starting point for defining our genes, which translate into different phenotypes or physical traits. In today's workshop, we will be talking specifically about how scientists are able to edit DNA to bring about desired phenotypes. But first, we need to understand what exactly is genetic modification. We also need to understand the molecular mechanisms for how mutations and modifications in an organism's sequence can lead to major consequences in an organism's structure, functions, or its phenotype. We will practice this through an activity called Color by Codon. Once we understand the fundamentals of how genetic modification can occur, we can explore a specific revolutionary technology that scientists are using to edit DNA to help cure diseases, produce more nutrient-dense and hardy foods, and much more. This technology has been named CRISPR-Cas9.
So to start us off, what exactly is genetic modification? Well, to put it simply, it is when DNA is edited in some way, and that brings about a change in the physical characteristic of a cell or tissue or organism as a whole. Now, genetic modification can happen via random mutations, like what underlies big important processes like evolution, or genetic modification can happen more intentionally, like the way that humans have bred specific dogs together to bring about desired traits like droopy ears or a squished nose. Now, what we are going to be learning about today is how scientists have figured out ways to modify genomes or DNA extremely specifically and selectively to bring about very precise phenotypes. And as we move through this workshop, I want everyone to keep in mind some ethical questions that may arise as this powerful technology is becoming more and more available. So to start us off thinking and moving, let's everyone take 1 minute and go find an object in your house that you think might be genetically modified. And as you come back, either type in the chat what you thought of or what you found, or if you want to turn on your camera and show us the objects, and we'll take a couple minutes and discuss them. We kind of predicted that a common thing that people would bring would be crops like fruits, vegetables, or other plants, but theoretically, you could genetically modify many different kinds of, beautiful orange fruit, good stuff, you guys. You're definitely along the same train of thought that we were when designing this workshop. So our first question, why do you think that scientists would want to genetically modify foods? Let's stick to foods because it seems to be the most common thing that you all thought about. Why would scientists want to modify the genome of these crops? Does anyone have any ideas? To make it mass production, that's a great inference because the world's population is growing a lot. To make bigger foods to help us feed more people, maybe to taste better. Exactly, to have more desirable traits. And so our next discussion will involve how does this happen on a molecular level? So when we talk about genetic modification, What we're really talking about is mutation, or the concept of changing the DNA in order to bring about a change in the physical characteristic, like I said earlier, of a cell or a tissue or crop as a whole. So we're first going to touch on the central dogma of biology, which is the fact that DNA in our cell nuclei gets converted to an RNA template that serves as a type of message and provides a template to build proteins, which are the building blocks of our cells and tissues and organs. Now, it is changes at the level of the DNA where the mutation happens, and that is what results in different proteins. And DNA mutations can happen in various ways. One possible way is in the enzymes that copy DNA templates when cells are dividing, like when we're growing, or if you have a wound that your skin needs to heal, those cells need to divide and copy the DNA template. So if you have a mistake in that replication, then you could have a mutation. Another way that mutation can arise is by DNA damage. So like what happens when you get a sunburn, it's the UV radiation damaging our DNA. Or if someone smokes a cigarette, that harmful smoke can damage the DNA of the cells that line our lungs. So now zooming into the molecular structure of DNA, well, DNA is made up of 4 different nucleotides: thymine, adenine, cytosine, and guanine. And these nucleotides are read as a template in groups of 3s called codons. So when you look at a DNA template, really the functional unit of it is the codon, so a group of 3 unique amino acids, and they can indicate different instructions, like the TAC you see here is a start codon, and the ACT is a stop codon, saying start here and stop here for the RNA template that will become a protein. And the one in the middle will indicate a specific amino acid, which is a building block of the protein. So if you think about how important this unit of 3 is, any mutation that disrupts the unit of 3 will have a big consequence for the resulting protein. So our first example of mutation is a point mutation. So that is when one specific nucleic acid is replaced by another. So in this case, the start and stop codons are the same, but that middle amino acid would be changed, would be a different amino acid. Our second example of a type of mutation is an insertion. So an insertion is when there's an extra nucleotide that is added into the DNA template. And this is classified as a frame shift mutation because now that you're introducing an extra nucleotide rather than just replacing one, it is affecting the groups of 3s, the codon functional unit. So in this case, the start codon is the same, but the next codon, taking both mutations into account, would be GTC. And the next codon would now be different as well. It would be TAC. So not only is there a change where the insertion happens, but in every codon thereafter. Our last example of a mutation is a deletion, which is also classified as a frameshift mutation because it is again affecting the functional codon unit of 3. In this case, a deletion in the stop codon would cause the following nucleic acid to be read in that codon unit instead. So instead of having an instruction to say, stop making the protein here, the protein would be longer because you've now lost or mutated that stop codon. Now, to reinforce what can sometimes be tricky concepts, we're going to do our next interactive activity led by Morgan, the color by codon.
Great. So now that you are starting to understand a bit more about molecular mechanisms of mutation, let us see this in action through a color by codon activity. In this activity, we will draw and color the original phenotype of a flowering plant, as well as the mutated phenotype of a flowering plant, using the instructions provided by its RNA sequence of codons. So from your materials that you have received, you will pull out the color by codon packet. So it should look something like this. And from this packet, you will see a set of instructions for the activities, as well as 2 activity sheets. So I want you all to grab sheet 1, which is titled Original Genotype and Phenotype, and grab your crayons so that we can actually use those to start drawing and coloring on the sheet. But before we start actually working on the sheet, let's explore it a bit more. So we can go to the next slide. So yep, that's the sheet that we want. So at the bottom of the page, which we'll see on this next slide, you will see the DNA template for the flower and the replicated RNA instructions. The RNA instructions are what you will be interpreting to draw the different phenotypes or features of the flower. Then on the next slide, we see that on the left-hand side of the paper, to interpret what the RNA instructions mean, we will need the codon instructions. And these instructions will help you read and interpret what each of the codon in the RNA instructions means. Then we have the right side of the page, and you will see on the right side of the page that there's a grid here. This is where you will actually be drawing the flower. So now that we have explored the page a bit, let's actually start drawing the flower's phenotype. So on this next slide, we have the first part of the RNA instructions circled. So we circled that first codon, and we see that the first codon is AUG, and AUG is a start codon. And this is pretty much the starting point for RNA translation. So to double check to make sure that AUG actually means start translation, we're going to go up to the left side of the page where we have the instructions, the codon instructions, and we will find the instructions for AUG. And we see that the instructions give you permission to start translation. So this is great. This means that we can go to the next codon in the RNA sequence, which happens to be CAG, as we see circled on the slide here. Again, after we see CAG, we go to the codon instructions and find out what CAG means. And so everyone should be looking at their paper, finding CAG in the instructions, and reading what you need to do, what you need to draw, or what you need to color. So once you know, use the gridded section of your sheet to draw or color the flower based on those instructions. And pretty much what you're doing is you're going along this RNA instruction strand and drawing the flower until you hit the stop codon, which is the codon that tells us to stop RNA translation. In this case, the stop codon is UGA. So we want you guys to take a few minutes to draw and color the flower to find out what the original phenotype of this flower is based on the RNA instructions. So go ahead and start coloring and drawing. And just a little pointer I like to make is, as you can see on the slide, we've circled each of the letters into groups of 3 to indicate the codons. And this can help you more easily read the RNA instructions so that you can go quicker. So I definitely feel free to circle. I've definitely circled on my page as well the codons to help me figure out what I need to be doing. And if you have any questions, please feel free to unmute yourself or post in the chat. Let's see. So some of you might still be drawing it out. Some of you might be done. So let's have everyone hold up their pictures of what they drew on their sheets. So you can see this is what I drew based on the codon instructions. And we see it up on the slide as well. So we see that with the original phenotype here, that this flowering plant has 2 flowers. It also has a couple of leaves. It has a branching root system, things like that. So keep this original phenotype in mind as we move on with the activity. So now that we know what the original phenotype of the flower looks like, what would happen if a mutation or a genetic modification occurred in the flower's DNA? What would the flower's new mutated phenotype look like? To answer these questions, let us move on to sheet 2 of the color by codon activity. And this sheet 2 is called Genetic Mutation and New Phenotype. And we see this sheet circled on the slide. So everyone should pull that one out. I have it right here in front of me as well. Okay, so pretty much with this sheet, as we see on the next slide, is pretty much the same as sheet 1, except now we will be using the mutant RNA instructions to determine the phenotype of the flower. Do exactly what you did before with sheet 1, but pay close attention to those 2 mutations that we see on the mutant RNA instructions. We'll give you all a few minutes to draw out the mutant phenotype on this one as well. Again, feel free to circle the codons on your sheet to help you figure out what codons you should be reading, especially since we see the point mutation in there and then the frame shift insertion in there. You might see that some of your codons have shifted because of those mutations. So keep that in mind. And again, if you have any questions, please feel free to put them in the chat or unmute yourself. So we should start to see some differences appear in this new drawing that we have for the genetic mutation. So you all can continue to draw as we move along, but this should be what the mutant phenotype looks like. And can anyone point out what new features we have in the mutant phenotype that we didn't have in the original phenotype? We can probably go on to the next slide so they can see the original phenotype and the mutant phenotype side by side. So what differences are we seeing between the mutant phenotype on the right and the original on the left? New petals and that black thing, yeah, the thorns on it. Yes, we are seeing some of those differences. There are more leaves on the extra flower. The stem length has changed. Yeah. So why do you guys think, for example, why would it be advantageous for a flower to have a mutation where thorns start to appear? What is advantageous about having thorns on a flower? There's a hint on your sheet. So if you look at the sheet, yes, protected from herbivores, yeah, as well as the insects as well. So if you have an environment where insects have become more prevalent, the flowers that actually have the mutation for thorns, where they start having thorns appear in their phenotype, might actually do better in that environment because thorns are a protective barrier. They help protect the flower from those insects getting to their stem and their leaves and munching on them. So thorns can actually be very advantageous. And so not all mutations are a bad thing. Mutations can actually bring about these phenotypes that are actually very, very helpful for the organism. So moving on, there are definitely people out there, like scientists, who are very, very interested in genetic modification. So I want to ask the question of how can we use genetic modification to help people and the world? And we've kind of visited this a little bit at the beginning. Cheaper products being made for low-income people. Yeah. Some of you also mentioned at the beginning that, yes, it can help prevent sickness, increase milk production. Yeah, so it can be used in the medical field to help people. It can be used in the agricultural industry, things like that. So on the next slide, we have an example of this. Just like we can change the phenotype of the flower, like we saw in this activity, scientists can change the genetics of crops to have advantageous phenotypes, like making them last longer on the shelf or making them drought resistant. So what is the technology scientists can use to actually change DNA? And we are going to move on to our next activity to answer that very question.
Okay, hello everyone. Before we do, we're going to go over what is CRISPR. So CRISPR-Cas9, often shortened as CRISPR, is a biotechnology tool that can be used to edit our DNA or DNA of other organisms and cells really cheaply and quite quickly. So what exactly is CRISPR and how does it work? So the first part of the name CRISPR, or cluster regularly interspace, refers to the part of the bacterial DNA that is spaced evenly. So there's a gap between this repeated sequence. And these regions are spaced that are interspaced are also palindromic repeats. And I wanted to ask you guys, what are palindromes? Can anyone know, or can anyone tell me what a palindrome is?
So we have dog and God. We have 101. We have race car, 2332. Things like that.
Yeah, they're hitting the mark. Great example, guys. So definitely. So palindromes are words that can read the same forwards and backwards. And yeah, 101, that's it, and dog and God. So, or I've written some examples here on the slides, like Hannah, or not in a jar of tuna, etc. So very good. So aside from these palindromes, what else do you find in this CRISPR system? So between these CRISPR regions, we have spacers, and these spacers, we find DNA sequences that are unique. And these sequences are actually matched to viral or bacteriophage DNA that have previously infected the bacteria. So in a sense, it's like a type of history of previous infections that the bacteria uses to recognize, oh, I've been infected by this pathogen in the past. Well, aside from this, CRISPR also has Cas9 proteins, or Cas-associated proteins. And what these proteins do is that they act as helicases, meaning they unwind DNA, and they also act as nucleases, meaning that they cut DNA to cut that DNA, that invading DNA, and keep a record of it. So why exactly do bacteria have this CRISPR system? How is it beneficial to them? So bacteria use CRISPR as a type of immune system to protect themselves from bacteriophages or viruses. So preventing these viruses from inserting their DNA and making many copies of themselves. So to counter that, the bacteria takes a little piece of that viral DNA sequence and inserts it into its own genome, or in the CRISPR array, seen there in the colored boxes. So the CRISPR system becomes like a history of past infection. So if the same virus tries to infect the bacteria, the bacteria will know, it will detect it and destroy it. Similarly, if this is a new pathogen that the bacteria has never seen before, the bacteria can take a little piece of it and keep a record of it in case this same pathogen tries to infect them again in the future. So this is a pretty neat immune system, a little different from ours, but still pretty neat. So, but better yet, we can use this CRISPR system to edit our own DNA. So since CRISPR can use to target a specific DNA sequences, we can design a guide that targets a segment of DNA of our own choosing. And the Cas protein will make a cut where we want it. Oh, sorry. Oh, we went so fast, I'm sorry. The Cas protein will then make a cut where we want it, and it can delete a DNA, it can delete a gene, or we can insert nucleotides, and we can insert like a new DNA fragment that could potentially even fix a mutation. So the implications for CRISPR in medicine are quite big. So to understand a little bit more how exactly Cas9 and CRISPR is working, we're going to do our own CRISPR modeling.
All right, guys. So if you could just pin me, we'll get started with this next activity. Okay, so in the second packet, you should have gotten a paper model of CRISPR-Cas9. And so now we're going to talk about how this discovery in bacteria ended up becoming such an important tool that scientists use today. And so when we build this model, we're going to show you how the CRISPR-Cas9 finds a specific region of the genome to cut, how it cuts it, and then how the DNA damage repair mechanisms that occur to fix that broken DNA actually can alter the DNA to have a new sequence and therefore change the code of the genome or change the sequence of the genome. And so in step one, I just want you to all get this piece of paper with this jelly bean looking thing on it. So this is actually approximately what CRISPR-Cas9 actually looks like. So I just want you to cut that out along the dotted line. And then you'll also see there's a couple tabs here, and I also want you to cut those out as well. And I'm just going to give you about a minute to cut it out. It doesn't have to be perfect. So what you're going to do is take your Cas9 protein that you've just cut out and take your tabs, and you can see there's a short edge here and a long edge here on top. And there's these little boxes on your CRISPR-Cas9 protein that are outlined there. And I just want you to tape the short edges, so just the small sides within those little boxes that are on the Cas9 protein. And try not to use too much tape because we want to be able to actually insert other pieces of paper into these tabs later. Okay, so hopefully many of you have your tabs now taped on to your protein. And so the next thing that we're going to do, we can move on to the next slide. So we're going to take the next sheet from this activity, and you'll see that there's some DNA here. There's this thing here called the guide RNA, and then these random nucleotides. And I want you to cut all three of these things out. And just real quickly, it doesn't have to be perfect. And then we'll have all our materials, and then we can assemble our model. Okay, so the first thing that we're going to do is we're going to assemble the CRISPR-Cas9 complex. And so what we're going to do is we're going to take our CRISPR-Cas9 protein, and we're going to take our guide RNA, this little hairpin looking structure. And what we're going to do is we're going to link up the guide RNA to the CRISPR-Cas9 protein. And there's a picture of this on the slide, and I'll show you here. So you're going to lift up one of your tabs and slide the guide RNA underneath it. And then this little hairpin structure goes into the top tab right here. So it should look like this. And so in real life, the guide RNA and the CRISPR-Cas9 protein are physically linked up together. And you notice that there's two different colors on this guide RNA. There's the blue part, and then there's a red part. And so the blue part is the part of the RNA molecule that physically links up to the CRISPR-Cas9 protein. And we've shown that with these tabs. And the red part, you'll notice it has a sequence on it like DNA, but we know this is an RNA molecule, right? But this is how your CRISPR protein is actually going to find what region of the genome it wants to cut, is because this red part will actually have a complementary sequence somewhere in the genome that tells our CRISPR protein where to go. And so now if we go to the next slide, we're going to model how the CRISPR protein actually finds the DNA. So now this is where our DNA comes into play. So this is, we'll just think of this as DNA just existing in your cell. You've got billions and billions of base pairs in every single cell in your body. Well, not every single cell, but that's a topic for another day. And you have this CRISPR-Cas9 protein, and it's just floating around in the nucleus of the cell, trying to find whatever part of its guide RNA matches to the DNA. And so eventually it finds it. So what we're going to do is take the DNA, and we're going to slide it underneath that tab where the guide RNA is also in the protein. And you're just going to slide the DNA in there until it matches up perfectly with that guide RNA. And this is how the CRISPR-Cas9 protein actually finds which area in the genome it's supposed to cut. And so now we're going to model the cutting. So if you look along your DNA here, you'll actually see that there is this black double strand or black line here, dotted line. And what I want you to do is take a pair of scissors and cut along that line. And don't cut the guide RNA. The guide RNA doesn't get cut, just the DNA. And you should have something that looks like this with your broken DNA now. So does anyone know what the cell does when it has broken DNA? Does it just let it live its life, or does it try to do something with it? Any ideas? Tries to fix it. Yes, exactly. Exactly, DNA repair, perfect. So now we're gonna model what happens during DNA repair. Mariella briefly touched on this, that there's this fancy form of DNA repair called non-homologous end joining. I'm not gonna bog you down with the details of that, but what you need to know is that when a cell attempts to repair DNA, oftentimes there can be actually mistakes made when it repairs it. And so the sequence that was in the DNA before will maybe not be the same afterwards, after the repair. And this can happen by taking away nucleotides here, by adding some. And what we're going to do is model the latter. So we're going to add nucleotides into this double-stranded break. So this is the part where our random nucleotides come into play. So you're going to grab your tiny little random nucleotides, and I want you to tape them in between the DNA so that you have essentially modified the sequence of the DNA itself. And now your DNA looks something like this. So it's been modified. The sequence is different. And if you think back to your color by codon activity that we already did, how do you think that changing the DNA sequence will perhaps affect the phenotype, like the protein that this gene should give rise to? Right, so it's gonna change the DNA. And what does the DNA ultimately give rise to? Remember, we talked about the dogma of biology. Someone in the comments gave a good answer. So it could potentially change the protein, right? So just like earlier, when we made some mutations in the flower that gave rise to a different looking flower, right? So same thing here. We've added some nucleotides into this DNA, and now maybe it'll give rise to a different protein.
So how can we use CRISPR technology for the benefit of humankind? So I want you guys to share in the chat or say or unmute yourselves. Give me some pros and some cons that we have, that you guys have studied yourselves here, or that we have mentioned in our presentation today. I'll start off. Like pros, like we had that slide on make vegetables. So making, and someone in the chat actually mentioned making bigger foods or better foods. So pros, we could potentially cure diseases by changing a mutation or making better produce, longer lasting or pest resistant. But some of the cons is that CRISPR technology is relatively new, so we don't know the consequences, the long-term effects that using the technology can have. So we need to first understand it a little more before we can use it in medicine. And there may be ethical issues surrounding CRISPR, because since you can modify any technology, you could use it for potentially ethical issues, like maybe modifying babies to choose the characteristics that you want to have in your child rather than what they would naturally be. So we have to be cautious of using the technology, but this technology would also allow us to have so much goodness in the future, especially if we could cure diseases. So going into that, so how can we use CRISPR for the benefit of humankind? But before we answer that, we have to make sure that the technology is safe. And the way that we, in the USA, the way we do that is we have a regulation, a regulatory body called the FDA, and they make sure that medicines or medical devices are safe for us to use before they're given to humans. And they require extensive clinical trials. So what are clinical trials, or have you heard of any recent clinical trials? Doesn't have to be CRISPR related. Have you heard recent clinical trials in the news? it could be for this very hot topic right now that's pandemic related.
Yep, the chat says COVID vaccine. Philip said that.
Yes. So actually, they have been many clinical trials with trying different vaccines, and one of the vaccines was actually removed from because side effects. But anyway, but the clinical trials allow us to feel safer when taking a medicine, and it's tested with millions of people eventually. Like the COVID vaccines have actually been tested with millions of people and have been proven to be safe. So yes, so there is a rigorous process that has to be followed before we can use this treatment. And I'm just going to show you some of the ongoing CRISPR clinical trials using this technology to cure different diseases. And one is a rare form of blindness, like using CRISPR to fix that mutation. Another one is actually engineering human immune cells to attack cancer cells. And if they attack to attack and recognize and kill cancer cells, like as a potential cancer therapeutic. So this means that CRISPR could really do wonders for human health. So with that, we go here. Who invented it?
And so finally, we see that the two women who came up with this revolutionary technology are Emmanuelle Charpentier and Jennifer Doudna. And they actually received the Nobel Prize in Chemistry in 2020. And so with these genetic scissors, the life sciences have entered a new era, and it's a huge benefit to humankind. And it shows us that not only are women in science capable of making a change, but women are currently revolutionizing all different fields of knowledge at this very moment. And here you can find links to some interesting videos that discussed what we have learned here today, including a TED Talk on CRISPR technology by Dr. Doudna herself. Also, if you're interested in learning more about what SACNAS is up to, you can find the link to our Facebook page here, and SACNAS stands for the Society for the Advancement of Chicano/Hispanics and Native Americans in Science. Thank you so much.
We are all members of SACNAS, and thank you for participating in our workshop today.